This is Ritik sir, your biology subject matter expert from memoneet. In this video, we will be discussing about the principles of biotechnology. You know what do you mean by genetic engineering? So, genetic engineering is actually a technique to alter the chemistry of the genetic material. So, it could be either DNA or it could be either RNA, and to introduce these altered genetic material into where a host organism and thus change the phenotype of the host organism. That means, so you're gonna add up or you're gonna introduce, okay, the altered DNA or desired DNA or RNA that you require into the host organism, so that there you will be expecting the change in the phenotype, that physical appearance from the next generation of the host organism. And from that next generation, whatever the desired product that you wanted or desired characteristic changes that you wanted, so will be expressed. So one thing you have to know, that maintenance of sterile, that is microbial contamination free. So ambience in the chemical engineering process to enable growth of only the desired microbes, eukaryotic cells in large quantities for the manufacturing of the biotechnological products like antibiotics, vaccines, enzymes, etc. That means whenever we want a desired, okay, organism, so one thing that we need to maintain is maintain a sterile condition. So a condition where there is free of microbial contamination. Okay. Then with the help of the chemical engineering process, we will enable the growth of only desired microorganisms. Okay. Then in a large quantity, we'll use eukaryotic cells to manufacture them, like such as a antibiotics, or it could be the vaccines, or it could be the enzymes, and many. So you know this making of curd or bread or wine. So which are all microbe mediated process. That means microorganism plays a very important role in this. So this could also be a thought as a form of biotechnology. And another thing, for example, in vitro fertilization leads to a test tube baby. This is one thing. Then synthesizing a gene and using it, developing a DNA vaccine or correcting a defective gene. So this all thing is actually a part of biotechnology. So whatever the vaccines that we are producing or any gene that we are correcting or any new gene that we are inserting. So this everything is actually a part of biotechnology. So there is one institution that is called as European Federation of Biotechnology. So this is actually an institution which has given a proper definition of a biotechnology that incorporates both traditional view and modern molecular biotechnology. That means combining both the traditional view of biotechnology and present modern view of biotechnology. And it defines that the integration of natural science and organisms, cells, parts thereof, and molecular analogous for products and services. So one thing that we need to know is the sexual reproduction has more advantages over what? Asexual reproduction. So the former provides an opportunity for various formulation of unique combinations of genetic setup, some of which may be beneficial to the organism as well as the population. Whereas coming to asexual reproduction, it prevents the genetic information While sexual reproduction, what happens? It permits the variation. Okay. Then we have traditional hybridization process. So which is used in plants and animal breeding, very often leads to what? Insertion. That is multiplication of undesired gene along with the desired gene. The technique of genetic engineering, which includes creation of recombinant DNA and use of gene cloning and gene transfer. So these all things will overcome the limitation and allow us to isolate and introduce only one or a set of desired gene without introducing undesired gene into the target organism. So one thing that here we need to know is how. For example, if you take a piece of DNA, it would not be multiply itself, right, in a progeny cell of an organism. But when a piece of DNA is get inserted into the genome of the replication, so it may multiply and be inherited along with the host DNA. So if you take a DNA and you expect it to multiply, it won't. Rather, if you introduce into a host DNA where this multiplication will happen, so it may multiply and it also gets incorporated with the genome of the host DNA. This is because the aligned piece of the DNA has become now a part of a chromosome which has the ability to replicate now. Okay. So, and this has what? Ability to replicate. So, one thing is in a chromosome, there is a specific DNA sequence here which is actually called as origin of replication, where it is, yes. So it is actually called as origin of replication. So which is responsible for the initiating what? The replication. Therefore, for the multiplication of any aligned piece of the DNA in organism is need to be actually what? A part of chromosomes, which has a specific sequence known as origin of replication. Thus, an aligned DNA is linked with the origin of replication so that this aligned piece of DNA can replicate, okay, so that it can replicate and multiply itself in the host organism. And this can also be called as cloning or making multiple identical copies of a template DNA. Just know it. Now, the construction of the first recombinant DNA embedded for the possibilities of the linking a gene encoding antibiotic resistance with the native plasmid. So, for example, here they have taken Salmonella thymorium. So here, what actually happened is that was the first recombinant DNA which they have taken, right? So next one is Stanley and Herbert. So they accomplished this isolation in 1972 by isolating the antibiotic resistant gene. Okay. So by cutting off the piece of DNA, first they have cut the piece of the DNA from the plasmid, which was responsible for the configuring what? Antibiotic resistance. Then the cutting of DNA at one specific location was only possible with the discovery of so-called molecular seizures. So these molecular seizures are actually called as what? Restriction enzyme. So the piece of DNA was then linked with the plasmid DNA, and these plasmid DNA, it acts as vector to transfer the piece of DNA attached to it. So you probably know that mosquito attacks is a vector, right? So for malarial parasite into the human body. In the same way, okay, just in that way itself, the plasmid can also be used as vector to deliver and align piece of DNA into the host organism. So the linking of antibiotic resistance gene with the plasmid vector becomes possible with the enzyme. So that enzyme is actually called as DNA ligase enzymes, which actually acts on cut part of DNA molecules and join their ends. It acts as glue actually. Okay, that we need to know. So this makes a new combination of the circular autonomously replicating DNA created in vitro and is actually called as what? Recombinant DNA. And when the DNA is transferred into the E. coli, a bacterium which is closely related to the Salmonella, it could replicate using a new host DNA polymerase enzyme and make a multiple copies. And the ability to multiply copies of antibiotic resistant gene in E. coli was called as cloning of antibiotic resistant gene in E. coli. So now the thing is you have infer that there are three basic steps in genetic modification of an organism. So the first one is identification of DNA with a desired gene. The next one is introduction of the identified DNA into the host, right? And maintenance of introduced DNA into the host, then transfer of the DNA to its progeny. So, what are the tools for recombinant DNA technology now? So, one thing we understood is restriction enzyme, then polymerase enzyme. We should have ligase, a vector, then the host organism. So, restriction enzyme is used to cut the piece of DNA. Polymerase enzyme is used to incorporate it. Ligase acts as a glue to stick the aligned DNA with inside the this thing vector. So we should have an appropriate vector so that we can insert into the host organism. So we will know what is restriction enzyme. Okay. In the year 1963, the two enzymes were which are responsible for restricting the growth of certain bacteria like E. coli was isolated. So one of this added what? A methyl group to the DNA, while the other actually cut the DNA. So the later it was the which like cut the DNA, we normally call them as restriction endonuclease. Okay. So the first restriction endonuclease that is hind 2 also cut the DNA at one specific part, as you can see here. So, to its one specific particular point by recognizing the specific sequence of six base pair, okay, of six base pair. And this specific base sequence is actually called as recognition sequence of the hind 2. Beside IND2, so today we have almost 900 restriction enzyme that have been isolated from over 230 strains of bacteria, each of which recognizes different recognition sequences. So the conversion for naming these enzyme is the first letter. That means how do you name this hind2 we got or some other things we name, right? How do we name them? So the naming of this enzyme is the first letter of the name comes from the genus, right? And the second letter that you are talking, yeah. And the second letter that we are talking comes from the species of the prokaryotic cells from which they have been isolated. Say if you have taken an example of E. coli, so we have E. coli here. So when I'm talking about an E. coli, it comes from E. coli cell, that is Escherichia coli. So here we have RY13. So eco RI, where R represents or it is derived from the which strain it has been taken. And then the 13 represents the normal number, follows the name indicates the order in which the enzyme were isolated from the strain of the bacteria. Then we have restriction enzymes belong to the large class of enzymes. So they are actually called as nuclease. And there are two kinds. These are of two kinds. One is exonuclease and endonuclease. When I'm talking about an exonuclease, removal nucleotides from the end of the DNA, whereas an endonuclease makes a cut at specific position within the DNA. And each restriction endonuclease function by inspecting the length of the DNA sequence. And once it finds its specific recognition site or sequence, it will bind it to the DNA and cut each of the two strands of the double helix at specific point in the sugar phosphate backbone. And each restriction endonuclease recognizes a specific palindromic sequence, okay, in the DNA. So what is palindromic sequence then? So palindrome and DNA is a sequence of base pair that reads same on the two strands where origination of reading is kept as same. For example, if you just look here, GAATTC. So from right to left, from 5 dash to 3 dash, if you read the same thing you're getting. And if you read from left to right, that is 5 dash to 3 dash again. So you are, sorry, 3 dash to 5 dash. Again, you are seeing the same thing, GAATTC. So you have right. So this is what the palindromic syndrome is. Sorry, not syndrome, palindrome. Right. Next we have how do we incorporate that? Right. So we have a foreign DNA here. So with the help of certain restriction enzyme, we cut both the foreign DNA and one piece of vector DNA here with a specific point. Now we try to incorporate the desired chromosome of the foreign DNA into the vector with the help of ligase, okay, which joins the foreign DNA to the plasmid. So once the plasmid is ready, it is incorporated or it is transformed to the E. coli, okay, which is the desired organism, and cell division takes place. And in each cell division, you'll get the desired character that you were expecting of. So once it is done, there is another thing called separation and isolation of the DNA fragment. Like you got what you, how do you know like what is the desired one that you want to get for that? So the cutting of the DNA by restriction endonuclease resulting in the what? So that results in the fragments of the DNA. Okay. For example, we have the DNA here. So we are gonna cut that with the help of restriction endonuclease. So in that way, we have pieces of DNA here. These fragments can be separated by another technique, which is actually called as gel electrophoresis. Since the DNA fragments are negatively charged molecules, they can be separated by forcing them moving towards the anode under the electric field through the medium or matrix. And the most commonly matrix that we use is gel agarose, which is natural polymer extracted from the seaweeds. Okay. And the DNA fragments separate according to their size and weight. Hence, the smaller the fragment size, further or farther it will move. So that is what we need to understand. That is, see, the smaller will move actually lit farther when compared to the larger size. So this is all about what do you say that? A basic introduction of biotechnology and application. In the next video, I'll cover the entire chapter. That means the remaining part of the chapter. Till that, if you have any doubt, you can, comment. You can contact me through the comment section or through the Telegram channel. Don't forget to like, subscribe, and share with your friends. Thank you.